Now, it's my honor to introduce the moderator, Dr. Sheila Singh, who will guide us through the event. Dr. Singh is a professor and head of neurosurgery, pediatric neurosurgeon at McMaster Children's Hospital, and the inaugural director of the Center for Discovery in Cancer Research, a CDCR, at McMaster University. She holds a Tier 1 Senior Canada Research Chair in Human Brain Cancer Stem Cell Biology and served as the founding director of the McMaster's Surgeon Scientist Program. Her lab applies a development neurobiology framework to the study of brain tumorigenesis, and she has published extensively with over 200 manuscripts in prestigious journals. Dr. Singh has won many awards, including most recently the 2024 Canadian Cancer Society Robert L. Noble Prize for Outstanding Achievements in Basic Biomedical Cancer Research. She's a fellow of the Canadian Academy of Health Sciences and of the Royal Society of Canada, and is a council member of the AACR. Sheila, thank you so much for helping me organize this event, and also for moderating the event. I'll turn it over to you now.
Thank you, Jai. Jai, thank you so much, Jai and team, for organizing this wonderful series. It really represents the spirit of our team because we are a very collaborative group of researchers and we are very interdisciplinary. And this is a kind of whole is the bigger than the sum of the parts picture. So I'm going to share my screen with you just to do a very, very brief introduction of, the background of how this paper came to be, and also a little bit about the subject matter of the paper, which is glioblastoma. So glioblastoma, I think, probably doesn't require a big introduction for this particular audience, but this is one of the most deadly cancers of high unmet need because our standard of care, which consists of maximal safe surgical resection followed by cranial irradiation and chemotherapy with the oral alkylating agent temozolomide, really only garners a 4 to 5% 5-year survivorship. And this number just has not moved for decades. And in fact, we're in a very unfortunate situation of having only a handful of approved drugs, even over 30 or 40 years. And that is simply because this is one of the most aggressive, clonally evolving and heterogeneous tumors imaginable. And so really, this is an area where we need to make huge headway. And arguably, you can see here on the right of my slide that you can see the primary tumor there that's treated in with the modalities that I've mentioned. Post standard of care, you have a scan that looks pretty good, but within a median 8 months, everybody relapses or recurs, and 96% of patients relapse or recur with a scan that looks like the one on the right. And the question that my team sought to ask is, why are we studying so much about the biology of the primary tumor when anywhere from 63 to 78% of the actionable clonal drivers of this tumor have evolved out by the time you get to recurrence? And why don't we understand more about the biology of recurrent glioblastoma, which is, after all, when patients are entering clinical trials and need those new targeted therapies or drugs? And so that's why our team assembled, and we were very lucky. We've had multiple sources of funding over the years, but we were seeded at the very beginning by a Terry Fox program project grant that brought together this group of very interdisciplinary scientists with the sole aim of trying to target this rapid evolution and the clonal heterogeneity in treatment refractory glioblastoma. And we wanted to do this with a very new approach, relatively speaking, in solid tumors, which is with novel and empiric immunotherapies. So we assembled our team of experts and gathered around the really lucky and excellent patient-derived samples that we get at McMaster University. Now at Hamilton Health Sciences, we have an incredibly hardworking team of neurosurgeons. They are actually the most clinically active neurosurgeons in the province from statistics I recently saw. And so they operate on multiple brain tumors a week, and every sample is sent to the Singh lab, where we've been able to develop very beautiful brain tumor initiating cell-based models of both primary and recurrent glioblastoma. And there you can see all of the different experimental paradigms that our experts brought to the table in order to do deep characterization, profiling, and really an aim towards target discovery. And at the end of the day, at the bottom of the schema, you can see our goal is to really collate all these different levels of data, from on DNA to RNA to protein and beyond, and really try to uncover novel targets that characterize recurrent disease. And this is actually the way that we wanted to frame our interactions. We thought, every one of us has a specific expertise that we can bring to bear. And what are we trying to do here? We're trying to go from basic discovery all the way to developing a new therapy for a patient. And so we built out this pipeline where every single one of us had an important role along the way, from target discovery, which I think in this case, Professor Jason Moffat is going to tell you a little bit about the discovery platform that really yielded this key discovery that underpinned this paper. Moving on to the second program there in the middle, where you can see that pillar involves all of our phenomenal therapy development experts, one of whom is here with us today, and that's Professor Kevin Henry, who is honestly one of the most creative and deeply knowledgeable antibody engineers I've ever met. And then, of course, on to Project 3, where we do some more testing. And, very often when you have a very large and diffuse program like this, you have to carve out projects from within that. And those projects are assigned to graduate students and postdoctoral fellows in our labs, and they work together in small teams of trainees. But it really takes one of those exceptional PhD students where, you thank your lucky stars that they came to your lab. And in that case, that was Dr. Chirayu Chokshi, who did his PhD within the framework of this amazing program. And I would like to call him probably like a super PhD student, because he didn't just learn the material within Singh Lab. He actually learned from every single collaborating lab that he had the luck to work with. And so he's kind of evolved into this super scientist with, an amalgamation of all of the knowledge in this program. So this program has already produced this was actually the first target that came out of our pipeline and was developed, and that is CD133. That's a story for another day, but it's just an example that all I wanted to tell you is we realized with the first hit, wow, this pipeline works. It's yielding actual novel actionable targets, and we can develop immunotherapy. So this was our proof of concept. But this is the paper that we're going to discuss today. And really all I want to do to share this with you is to say that we published this paper, and there's no doubt in our minds that for all of us, this was a huge win, but it was not easy or straightforward in any way. In fact, this paper took 7 years of work. And even though it found value in the international community at the end of the day, really the story was underpinned in this Nature research briefing that they published, where they show behind the paper, and this is where all the action actually happens, behind the paper. So just so you know, this was not a straight trajectory. This was not a smooth path. We fell into many manholes. We bumped our heads against many walls. We were rejected so many times. I think Chirayu was depressed at multiple points during his PhD. So this was not an easy path. And I'm telling everyone this because very often you see a paper like this, you don't realize it's 7 years of work by over 100 scientists. And we had to keep pivoting and getting ahead of the data in order to actually land on this beautiful story that ended up being published in Nature Medicine. So we want to tell you the story of that paper today, and we've brought some key team members to take you from the beginning to the end of this beautiful trajectory. And so with that, I'm going to stop sharing and introduce my colleague, Professor Jason Moffat, who is one of the world's most innovative and leading experts in functional genomics, Canada Research Chair of Functional Genomics, and currently the head of the program for genetics and genome biology at the Hospital for Sick Children in Toronto. And he did some of this work from where he started his career, which is at the Donnelly Center in Toronto, where I think they brought him to sort of establish and innovate all of the most cutting edge at the time shRNA platforms that evolved into CRISPR. So with that, I'll sign over to Jason.
All right. Thank you, Sheila, for that nice introduction. And thanks for the opportunity to provide some thoughts from my side of the table on this project and this publication. I'm going to step a little bit back in time before we started doing screens with Chirayu in Singh Lab in his really nice models of GBM, and tell you a little bit about why this was even possible. So I think most people know about CRISPR screens and probably RNAi screens before that. And so my lab was sort of key in developing the Toronto knockout library, the first version of that, as well as subsequent versions. And typically the way that these screens work is that you introduce a library of guide RNAs into a population of cells so that each cell gets the same guide RNA, and that guide RNA is represented in hundreds of cells in that population, and you allow that population to evolve, the guide will go and create an indel or an edit in a target gene of interest, and the resulting DNA repair outcome from that particularly indel typically is a either sort of a loss of function allele or a hypomorphic allele for that particular gene. So we can track those populations out over time, and then we can use deep sequencing to understand what genes are important for the growth of those cells over time. And the hardest part in all of this, actually, when it all started, was to analyze that data in a robust fashion. And this actually took, this goes back pre-CRISPR screens to RNAi interference. And I don't think people realize how important the RNAi field was at establishing methods for doing CRISPR screens, especially using lentivirus. And one of the key things that we helped to contribute to that field was an algorithm that allowed us to quality control for screens. And this was published by a former postdoc in my lab while he was there, describing this algorithm we call BAGEL that allowed us to make sure that essential genes, or at least the set of essential genes that we had defined at the time, were behaving properly in a CRISPR dropout screens, and that the reference non-essential set was also behaving properly. And this was actually kind of a bit of luck that the first CRISPR screening papers came out when we were trying to set that technology up. And it was clear that one of the screens that was published didn't yield any of the known essential genes in human ES cells. But if we use this kind of quality control metric, you can really establish good controls for screens. And we were able to do that. And this is shown here in a number of different cell lines. So we came up with this model called core fitness, or common essential genes, that we could use to start defining context essential genes. And you'll see why this is so important in the Nature Medicine paper in a minute. But that allows us to kind of anchor in the essential gene set to understand what genes could be important for a particular context or condition. And so we evolved the libraries, and that's when in talking with the Singh lab and Sheila, we ventured into this odyssey of screening GBM models developed in her lab. And so, as Sheila already mentioned, this really was spearheaded by an incredible PhD nurse in her lab, Chirayu Chokshi. And so what Chirayu and a former master student in my lab, David Tu, did was they thought it would be really smart to take primary GBMs and do a CRISPR screen in these models using standard of care. So either radiotherapy, temozolomide, or the combination. And the idea was that maybe we could get at some of the recurrent dependencies by doing these types of screens. So Chirayu and David spent a lot of time doing these screens. This was a lot of effort. And all of the screens actually functionally work quite well by all the metrics that we could look at the time. And we were able to look at conditional dependencies under these either radiation, temozolomide, or the combination. But between the two different models, nothing was really that consistent. So we kept asking ourselves, I guess, is this approach the right approach? And I think at the time, Chirayu was developing a recurrent model of GBM that was matched to one of these primary models that he had already screened. And he'll probably go through this in more detail, but he developed this recurrent model 972, and he characterized it very deeply, both functionally in survival curves in mice, as well as sphere-forming assays, and also genomically by doing exome sequencing. And the recurrent and primary lines were quite different. So at that point, he decided to screen the recurrent line. And this was, at the time, at least the first known matched primary recurrent pair that was screened at genome scale using CRISPR screens. And the idea was to look at this data and compare the primary recurrent effects and dependencies and see if there was anything either consistent with what we had seen in the temozolomide radiation and combination or not. And then he did something clever that I'll talk about in a second. But the first pass examination of the data did not clearly link any of these genes that are dependent in the recurrent model to the condition-specific dependent genes that he had found in his standard of care screens. But then going back, he then started doing a much deeper dive into existing data, including in the DepMap, as well as expression data. And this is where he found that there was a number of interesting genes that appeared to be fairly dependent in the recurrent model that he had screened that was matched with another one, as well as a different recurrent model that was screened that didn't have the primary matched set. But what was important was to compare this to the expressed genes. And then he came up with this list that we started to think about more. And then it became quite obvious that SOX2 is already known to be an important driver in GBM. But some of these additional genes were not really all that well characterized. And so I think he went after the second one, which made a lot of sense to me anyways. And then he went on to really start characterizing how PTP4A2 is an important regulator in this recurrent model and in other recurrent models. And so I will pass off the torch to Chirayu at this point.
And maybe just while Chirayu is setting up, I'll tell everyone that Chirayu is currently a senior scientist at Ensoma, which is a very cool biotech in South Boston. And Chirayu, thank you so much for coming today.
Of course. Thank you, Sheila and Jason, for introducing me and the work. And as Sheila mentioned, this pipeline includes so many field-leading scientists and their groups, and I was fortunate enough to tap into each one of their expertise and learn from them. And it all came together as this wonderful story that we hope to present to you today and push forward after this as well. So here I'll be focusing on how we validated our hit, which Jason mentioned was PTP4A2, and it led to multiple discoveries, both in terms of recurrent GBM biology, but it also showed us multiple directions in which we can target recurrent GBM in a therapeutic setting. So before I go into the data, I do want to share what is PTP4A2? I want to ask that question. And PTP4A2 is a phosphatase. So it removes phosphate groups from proteins as a form of post-translational modification. And so dual specificity phosphatase. So it does remove it from multiple amino acids. And not much was known about PTP4A2 when we started working with this a few years ago. But what we did know was that PTP4A2 was actually very important. It's part of a family that has 3 different phosphatases, 4A1, 4A2, and 4A3. And when 4A2, specifically the one that we were interested in, when in other studies, when mice PTP4A2 was taken away, it led to growth retardation, it led to impaired spermatogenesis, and defects in HSC cell renewal. And these are very significant phenotypes that tell us that PTP4A2 is indeed very important for mouse growth and survival. So, and we also knew from our prior work, as Sheila pointed out with CD133 and with others, that tumor cells have this intrinsic strategy that they're going to hijack something or hijack a protein or a pathway that is very important in development of normal mammalian cells and hijack it for the benefit of the tumor. And we thought this may be what we're seeing here. So when we look at PTP4A2 expression and we expand our horizon to not just glioblastoma, but also to gliomas, what we see is that the gliomas that express low levels of PTB42 have a better survival than the gliomas that express high levels of PTB42. And that tells us that, okay, PTB42 is actually important, that it distinguishes between disease within the heterogeneous beast that is glioma, between those that are very that lead to poor survival versus those that lead to better survival. And this kind of difference in survival tells us that we may be on the right track to discovering new biology here. Because when we started this work on PTB42, we didn't know anything. The field didn't know anything about what PTB42 does in glioblastoma. So we're excited to investigate that. So first, before we went into any kind of cool assays, we wanted to validate that our screening results were indeed accurate. And what we did is that we knocked out PTB482 using Jason and his lab's CRISPR technology from multiple patient-derived models developed in the Singh lab. And these are all patient-derived models of recurrent GBM. So from patients that have undergone standard of care and recurred with the tumor, and then we have developed models from those said recurrent GBM tumors. And when we knocked out PTB402 using multiple guides, we found that across patients, there is inhibition of self-renewal as assessed by secondary sphere formation, as well as proliferation of these tumor cells. So that tells us that our finding, not only do we validate what we saw on the screen in the same model that we screened, but also the finding that we have is actually applicable to multiple models. And that is important because GBM is a very heterogeneous disease. And to have consistent findings across GBM models is rare. After validating our work in vitro, we thought, okay, we can knock out PTB482, but and then see a phenotype that leads to recurrent GBM growth arrest. But can we do the same thing by inhibiting the phosphatase activity? Yes, the phosphatase activity is the main way that PTB482 interacts within the cell. So we reached out to Dr. John Lazo from the University of Virginia, who had developed a pan-PTB4A inhibitor, and it inhibits the phosphatase activity in particular. So this inhibitor called JMS053 inhibits the phosphatase activity of all three members of the PTB4A family, 4A1, 4A2, and 4A3. And this is a good time to tell you that these three members are actually very similar in amino acid sequence. And it's been proposed in literature that they might have redundant activities depending on context. So in a way, having a phosphatase inhibitor that targets all three means that any kind of redundant activity that may help rescue the cells from any kind of specific inhibition of PTB482 will be blocked by using this inhibitor. So first, we treated some patient-matched primary GBM and recurrent GBM cells with this inhibitor at different concentrations, and we also included neural stem cells as a control. And we found, as you can see in the red here, that the inhibitor worked really well in vitro at inhibiting the growth of recurrent GBM line, but it largely spared the primary GBM model. But more importantly, it spared the normal cells. So we don't want the inhibitor to have any kind of detrimental effect on normal cells, which is why we did this test in addition to others. So what we did find is that not only does the genetic knockout of PTB482 diminish the growth of this recurrent GBM model, but when we inhibit the phosphatase activity of PTB482, that also leads to the same phenotype. So that is great validation, again, for our work here. And then we expanded to other patient-matched models of primary and recurrent GBM. And here we found something interesting, that JMS053 is not only effective on the recurrent GBM model shown at the bottom, but it's also effective against the primary GBM model from the same patient. So this tells us that the biology that we discovered in the models that we screened with Jason's CRISPR libraries may be applicable not only to recurrent GBM, but also potentially to primary GBM. And this, again, showcases the heterogeneity among GBM patients, that something that we find may be applicable in different ways to other patients. But it's great to see that the inhibitor works in multiple recurrent GBM models and validates our genetic screening results. So then we expanded to this. Okay, so we know it inhibits the growth of these recurrent GBM cells, but does it actually inhibit the invasion of these cells? Because recurrent GBM is a highly invasive tumor, as I'll show you soon, looking at an MR image from a patient. And inhibiting the invasiveness of GBM is an amazing therapeutic approach to actually stop the spread of the localized tumor that may be accessible to surgical resection to becoming a beast that is no longer able to be treated by other therapies. So here we see that treatment of recurrent GBM cells with this inhibitor also inhibits the invasive capacity of these cells. And again, we repeated these studies in multiple models, multiple patient match models of primary recurrent GBM, and consistently found that when treated with GMS-053, the recurrent GBM models as well as the primary GBM models had inhibition of their invasion. So this is telling us that PTB482 is involved in invasion of recurrent GBM, in addition to being involved in the proliferation of these cells. So then we decided, let's go in vivo. Let's see if that when we give mice a recurrent GBM tumor, does this inhibitor also work in vivo? And what we found here is that almost 2 weeks of treatment of tumor-bearing mice in a xenograft model of recurrent GBM with this GMS-053 inhibitor treatment, that led to a modest increase in survival. And this kind of confused us a little bit, because why is such an effective molecule in vitro not translating in vivo? And then when we investigated this further using PAMPA assays and other assays to evaluate blood-brain barrier permeability, that's when we found that GMS-053 in its current form does not class the BBB. And that is very important for the treatment of brain cancer. And for this reason, we thought, okay, we need to do more work with GMS-053, and our collaborators, Dr. John Lazo and his team, are actively working on making GMS-053 more effective and better at permeating the BBB. But right now, we want to ask the question, can we get more insights into PTB4 activity in recurrent GBM? And will that reveal alternate therapeutic approaches? Because what we do know is that this axis that we are slowly discovering is a very good way of targeting recurrent GBM, but we just haven't found a therapeutic that is effective. So can we learn more about the mechanism behind this, more about recurrent GBM biology, and will that reveal alternative therapeutic approaches? So in order to do this, we perform phosphoproteomics, which is a way for us to understand what proteins are the PTB4A phosphatases interacting with and dephosphorylating, and will that tell us about which signaling axis is driving the growth of these recurrent GBM cells? So we use phosphoproteomics on the left here in collaboration with Dr. Yu Liu at the time, who was at the time at McMaster University, in order to treat both primary and recurrent GBM cells with either a control compound or the PTB4A inhibitor GMS-053. And by treating with both and looking at the alternative phosphoproteome in these two conditions, we can then quantify and perform functional enrichment on which phosphopeptides and phosphoproteins are being dephosphorylated by PTB4A proteins in primary GBM as well as recurrent GBM. And then see that, okay, what is happening in recurrent GBM that is so special as it relates to the phosphatase activity of these PTB4A proteins? And can we learn more about what that means for recurrent GBM biology? So when we took a close look at the differentially phosphorylated proteins, what we found is that there was a vast enrichment of proteins involved in axonal guidance. And of these proteins, one that stood out to be the most differentially phosphorylated in recurrent GBM compared to primary GBM from the same patient was ROBO1, or roundabout receptor 1. Now, ROBO1 is a very important protein during neurodevelopment, because what it does is that it allows for neuronal axons. It guides neuronal axons to where they need to get. And if I go back to the point that I made earlier in my presentation, that it is well known that recurrent or tumor cells in general hijack developmental processes for their own benefit. And we thought, okay, it seems like there's a connection between PTB4A and ROBO1, which we need to validate. And these two PTB4A is involved in invasion, ROBO1 is involved in axonal guidance and normal neurodevelopmental biology. That is there a connection here? And is that driving aberrant axonal guidance signaling in recurrent GBM? And what we found is that it wasn't just ROBO1 that was differentially phosphorylated in recurrent GBM, but also other proteins in that ROBO1 signaling pathway, including SLIT-ROBO GTP-associated proteins, CDC42, which is a very well-known driver of tumor cell invasion, as well as CDC42-associated proteins, all of which in a normal context drive actin polymerization, axon repulsion, and cell migration, all of which are important in development. But in the context of a tumor, these are all may all be hijacked for the benefit of the tumor. So validation of our phosphoproteomic data told us that indeed there is a connection between PTB4A inhibition and ROBO1, as when we treated recurrent GBM cells with the PTB4A inhibitor, we saw a decrease in ROBO1 levels. And when we look downstream of ROBO1 signaling, we also see a decrease in the active form of CDC42 that drives cell migration. And at the same time, we also looked through the phosphoproteomics data and found that beta-catenin was also being differentially phosphorylated in recurrent GBM. And beta-catenin, part of the Wnt signaling pathway, is well known to be a driver of recurrent GBM growth, or just GBM growth in general. And the Singh lab has published papers in the past before this on that said biology. So we then also validated that PTB4A is interacting with beta-catenin such that treatment of beta-catenin or treatment of recurrent GBM cells with the PTB4A inhibitor leads to increase in phosphorylated beta-catenin, which is a precursor to beta-catenin activity or Wnt signaling, canonical Wnt signaling in particular. And together, all of these mechanistic studies show that PTB4A2 is not just impacting ROBO1, but is having a multi-layered response where it's dephosphorylating ROBO1 as well as its downstream effector proteins that is allowing the tumor cell to invade. And at the same time, it's also dephosphorylating beta-catenin, and that is promoting tumor cell stemness and proliferation. And by inhibiting PTB4A2, by using the pan-PTB4A inhibitor, we were able to suppress all of these. And that showed up as a reduction in tumor growth invasion, self-renewal, and proliferation. And now we thought, okay, we are looking for an ultimate way of targeting this newly discovered pathway that no one has showed before in GBM and other cancers. Could ROBO1 be that answer? Can we find a way of targeting ROBO1 that would allow us to still leverage the biology that we learned about recurrent GBM, about this very great vulnerability in this tumor type? So to do that, we first assessed ROBO1 expression in tumor cells and in normal cells, because whenever targeting a cell surface protein, especially with any type of immunotherapy, we want to assess the expression within the tumor versus in normal brain to make sure that there is tumor enrichment of this protein so that it would be safe. Any kind of therapy that we do develop would be safe for patients. And we found when we reached out to our colleagues at Hamilton Health Sciences and gathered all of these different GBM tumor specimens, we found that ROBO1 is overexpressed in about a third of recurrent GBM specimens and almost a half of the primary GBM specimens. So this might be a tumor enrichment that occurs in primary and recurrent GBM. And as I mentioned earlier, GBM is a very invasive tumor. And this is a patient MRI from a patient that suffered from recurrent GBM and then passed and then donated their brain to the Singh lab, where we were able to analyze multiple different sections of this tumor. And here, we not only appreciate how invasive of a tumor GBM is, but we also can assess ROBO1 expression in different parts of this tumor. And it's well known that any single target in GBM is going to have heterogeneous expression within the tumor. And we see that here as well, such that section 3 or tumor region 3 here has very little ROBO1 expression, but tumor region 5 here has a lot of ROBO1 expression. But what we also can tell here is that parts of section 5 here, which represent the normal brain, have no ROBO1 staining. And that is important so that we can clearly see here that normal brain does not express ROBO1, and there is truly an enrichment in the tumor cells here for ROBO1 expression. So this may be a target for an immunotherapy. And an immunotherapy that we were particularly interested in and that we had experience with is CAR T cells. CAR T cells, or chimeric antigen receptor T cells, have shown great success in hematological malignancies and have been approved now. But they have yet to show success in the vast majority of solid tumor patients. And here, I'm going to go through this briefly. A CAR T cell is a chimeric receptor that leverages an scFv from an antibody that allows that receptor to bind to a specific antigen on the target cell. And then there's a transmembrane domain as well as signaling domains that allow that once the scFv binds to the target antigen, then leads to activation of the T cell. It sends T cell activation signals to the intracellular co-stimulation and stimulation domains, allowing for that T cell to be activated and then mount an anti-tumor response against the target cell that has just been identified. So using CAR T cells have shown success in a subset of solid tumor patients. So we know that success is possible. And then we wanted to ask the question, is ROBO1 a good target? And as I mentioned, CAR T cells have been tested in solid tumors. And this year alone, this is a subset of all the clinical data that has come out this year alone in CAR T cells for solid tumors. And there were three very seminal papers that came out earlier this year, around the same time that we published our work, showing that CAR T cells targeted against IL-13 receptor alpha, EGFR, or EGFR variant 3, modified CAR T cells here as well, that all of these are effective against a subset of patients, but not all patients. And what that tells us, knowing GBM biology and knowing that GBM is so heterogeneous, is that we need to expand the library of antigens that we're able to target to cover a larger fraction of that GBM. And going quickly through the largest ever phase 1 clinical trial in high-grade gliomas run by Dr. Christine Brown of City of Hope, targeting IL-13 receptor alpha 2 using CAR T cells. On the left here, what you're looking at is how long did the patients given this treatment survive? And if you see, the vast majority of patients actually succumb to their disease within the first 12 months after receiving this therapy. So this therapy isn't really working for the vast majority of patients, but in a few patients, it is working. And this is not specific to IL-13 receptor alpha 2, but indeed the case for other single-targeted CAR T cells. And the reason for this, the main main barriers to effective CAR T therapy is antigen heterogeneity in GBM tumors, but also combined with the immunosuppressive tumor microenvironment and the lack of CAR T cell persistence. And all of these need to be addressed. What we can address here is, or we can ask the question here, is that can ROBO1 CAR T cells add to this arsenal that we have against GBM to increase our coverage of what we can target in high-grade gliomas? And when we compared ROBO1 expression to existing targets that are being targeted clinically, we found that ROBO1 expression in patient-derived GBM models is comparable to other clinical stage CAR T cell targets, including IL-13 receptor alpha 2, EGFR, B7H3, and CD70. And now we want to ask the question, can we design ROBO1-directed CAR T cells, and can they be an effective therapy against GBM? And to answer this question and to go through the process that we took to evaluate this, I'll pass it on to Dr. Kevin Henry.
Thank you, Chirayu. That was phenomenal. And as we're passing over to Kevin, I just want to tell everybody that at this stage that Chirayu is sharing with you was the stage at which we first submitted the paper to Nature Medicine. And I think everyone would be interested to know that before the editor agreed to put this paper under review, she actually asked us to go back and prove that our target was actually comparable to other clinical stage CAR T targets before she would even consider reviewing the paper. So that was a kind of rush extra experiment we had to do to show that before we submitted. But now I'm really happy to introduce Dr. Kevin Henry, who is at University of Ottawa and NRC, when he undertook this incredibly exciting antibody campaign with us. So Kevin, thank you for coming today.
Thanks, Sheila. Thanks, Chirayu, and everyone for the opportunity to share our work. So to wrap things up, I'll be giving an overview of the discovery and preclinical development of the ROBO1 immunotherapy in the paper, which was an antibody-based CAR T cell. This was for sure a very collaborative effort and involved a lot of interplay between labs in Ottawa, Hamilton, and Toronto, which I hope will become apparent. So by way of introduction, I just wanted to start with a diagram from an older Paul Carter review. This has stuck with me because it illustrates so well, I think, how antibody-based immunotherapies are developed, or at least successful ones. So this would start either with the discovery of a new target or a desire to work with a clinically validated one. And then move to an antibody-based therapeutic concept for which design specifications can be identified. If necessary, reagents and assays are developed. And then we generate the antibodies, evaluate them in vitro and in vivo, optimize them if necessary, and then pick the best one to move it into preclinical or clinical development. So the main point to take away here is that antibody discovery is not really a one-time thing that begins and ends with picking the first antibody off the shelf that's just available. Not all antibodies will be fit for purpose. So, for example, some antibodies may not have appropriate properties for clinical development. In many cases, we hope we can optimize those. But in other cases, the entire antibody-based therapeutic concept or design specifications might be wrong, in which case we need to refine our hypotheses and start the whole thing all over again. Again, I hope some of these principles will become apparent in what I'll show you next. So, as is often the case in science, the interest in ROBO1 for treatment of GBM started actually, I think, before the fuller understanding of the PTP4A and ROBO axis was achieved. Sheila is smiling. What I'm showing here is a slide from a retreat, I think, in 2017, in which trainees from all the labs involved pitched their favorite targets to work on in the coming months and years. And I think at this point, the general role of ROBO1 in multiple different solid tumors, similar in sort of broad strokes to several other targets, was appreciated, but its specific link to recurrent GBM was just starting to come to light. So despite the early data being somewhat speculative, the hunch paid off. And while I wouldn't say groupthink is the best answer to all problems, I do think that teams usually steer the ship better in the face of uncertainty than any individual decision maker would. In any case, in these early days, we looked at ROBO1 from the same lens. We looked at any other protein to assess antibody drugability. So those criteria are shown on the right. They include things like the localization, structure, protein function, relative expression levels, homology with other related proteins, and with the orthologs in model organisms, and then the proposed mechanism of action, although for CAR T applications, that's fairly clear. So the first decision we made regarding ROBO1 targeting with immunotherapy was to use single-domain antibodies, also known as nanobodies, as targeting agents in light of the potential for a CAR T application, which would require an antibody fragment. So for any of you who are not familiar with these types of antibodies, homodimeric antibodies that have no light chains have emerged at least twice by convergent evolution. These are referred to as heavy chain-only antibodies in camelids. And immunoglobulin new antigen receptors in the sharks, respectively. So while the genetic mechanisms causing loss of the light chain are a bit different, the variable domains of camelid heavy chain antibodies and shark IgNRs, these are called VHHs and VNRs, have similar properties. They can fold and they can bind antigen autonomously in the absence of the remainder of the amino acid chain. So in the camelid system, the VHH domain has adapted to the loss of light chain pairing by the accrual over evolutionary time of polar and charge substitutions in framework 2 at the X-VL domain interface that make the surface less hydrophobic. So that's what's shown here. So we have the structure of a VHH on the left in cyan, and the structure of an FV fragment from conventional antibody on the right, with the VH domain in cyan and the VL domain in transparent green. So the CDR loops here are shown in white. And the biggest reason people like these molecules is because of their favorable stability and modularity compared with the fragments derived from conventional antibodies, such as scFvs, which often require extensive engineering efforts to arrive at a molecule with satisfactory properties for clinical development. So despite interacting with antigens with only 3 CDR loops instead of 6, antibodies can achieve comparable binding affinities and, like other small molecular weight proteins, may penetrate tissue a little bit more effectively. That's what's illustrated here. However, at least as 15 kilodalton monomers, they have very short serum persistence, but their half-life can be extended using a number of different strategies. Okay, so one of the challenges we saw right away with ROBO1 was its high degree of sequence conservation in camelids in humans as well as in mice. So for those of you who aren't aware, most antibodies for therapeutic indications are still produced by immunizing animals. There are several reasons for this. It does complicate things a bit in cases like this. What I'm showing here is the structure of the ROBO1 extracellular domain. So this ECD has 5 immunoglobulin-like domains and 3 fibronectin type 3 domains. Each domain's identity taking the human protein as reference is shown in the table here. So what you're going to see is that this is a fairly large ECD, nearly 100 kilodaltons. But what you can appreciate is that there are really only one or a few amino acid polymorphisms per domain, which we expect could be immunogenic in a llama, for example, since antibodies against the remainder of the protein will be removed by tolerance mechanisms. So, the workaround, if you were immunizing a mouse, for example, would be to make a ROBO1 knockout mouse. Although this is laborious and expensive, in a llama, you can't really do that. So, the other thing to note here is that even though the mouse and the human ROBO1 share 97% identity, the majority of the polymorphisms that distinguish the human and the camelid protein are actually not conserved between human and mouse. So although it might seem counterintuitive, it's actually a bit more likely in this scenario to get antibodies out of the system that bind human ROBO1 but actually don't react with the rodent protein very well. So anyway, we decided these risks were acceptable and went ahead with generating antibodies by immunizing a llama, constructing phage display nanobody library from the animal's peripheral repertoire, and then pulling up binders by panning. This was largely the work of an excellent RA and now RO at NRC, Martin Rizzotti, whose photograph seems to have gone missing from my slide here. So as we might have expected, the first attempt in this regard, that's shown on top here, campaign number 1, gave a panel of antibodies that all targeted roughly the same immunodominant epitope on ROBO1. The highest affinity of this set of antibodies was an antibody called MKRO20, which, as you can see in this isoaffinity plot here, had an affinity of about 12 to 15 nanomolar. And then subsequently, in a second campaign, whose rationale I'll explain in a moment, we pulled out another nanobody using a bit of a different strategy called XRO. And this bound human ROBO1 with an affinity of about 2 nanomolar and targeted a different epitope, as shown here in the sandwich ELISA. So only the pairs can detect in either orientation soluble ROBO1. Again, probably not surprisingly, given the near complete conservation across species, neither of the antibodies bound the ROBO1 first and the second Ig domains, which form the site of the SLT2 interaction surface. So here you can see a bit of a head-to-head comparison of the 2 lead molecules from the first and the second campaign. So again, MKRO20 and XRO. Panel A here shows a single cyclokinetic analysis of the interaction between the nanobodies and ROBO1. And the much slower dissociation of the XRO nanobody is pretty clearly evident. Panel B shows the reactivity of these antibodies across other members of the Robo family. So as you can see, we only see binding to ROBO1 rather than ROBO2, 3, and 4. Nor do we see any binding to rat ROBO1, which was maybe not a complete surprise based on what I showed you already. So we confirmed the specificity of these antibodies by flow against cell lines that were overexpressing or not expressing ROBO1. And it found in addition to binding, we could see their specificity in pull-down assays and tissue staining experiments performed in the Singh lab. So one of the nice things about the nanobody system is the ease of reformatting binders in various ways. So Chirayu, this may be a trip down memory lane. Right after we got the clones from the first campaign of the library, we performed some preliminary in vitro and in vivo testing by complexing the biotinylated MKRO20 nanobody with streptavidin. This gives a tetravalent complex that's large enough to pass renal filtration. And what Chirayu found was that the nanobody appears to decrease secondary sphere formation and cell evasion, but I think had less of an impact on the proliferation of recurrent GBM brain tumor initiating cells. Subsequently, he did orthotopically engraft recurrent GBM tumor cells in immunocompromised mice, and then delivered some local intracranial treatments of this tetramer. And although this was a small experiment, I think all 4 of the mice showed some tumor regression. So I think these early data illustrated that treatment with some ROBO1 targeted antibodies may mimic the effects of pan-P2P4 inhibition, again, which is shown, the diagram is shown here again. We aren't really completely sure of the mechanism yet, since this antibody doesn't actually target the site of ligand interaction. We speculated it may result in downregulation of ROBO1 expression from the cell surface. In any case, the studies of the effects of the naked antibodies kind of were put on the back burner because of our change in focus towards CAR-T application. As Chirayu already mentioned, based on some of the recent data that have come out of multiple labs now showing that local regional delivery of CAR-T modalities could be safe and maybe even effective in at least some patients with brain cancers, Sheila's lab went on to generate ROBO1 CAR-T cells, initially using the best 2 nanobodies from each of these discovery campaigns by cloning them into a second generation CAR backbone and transducing healthy donor PBMCs with the lentivirus packaged CAR construct. So this achieved transduction efficiencies of 80% or better, I think, after some optimization. And in vitro, these CAR-T cells, especially using the XRO nanobody, so on the left here shown in green, showed really potent specific lysis of different ROBO1 expressing recurrent GBM tumor cell lines, as well as an interesting early signal of efficacy against ROBO1 expressing medulloblastoma lines, that's shown here, MB002 on the top. In the panel C down here, you can see CAR-T activation profiles against matched isogenic ROBO1 CRISPR knockout recurrent GBM cells. This was a nice experiment Sheila's lab did to demonstrate the specific responsiveness of the XRO-based CARs. And above that is just a short video, which has now stopped playing. I'm not going to start it again. So this was an InKSight co-culture assay in which recurrent GBM cells are red, dead cells are green, and the CAR-T cells are in gray. And what you saw, if you were paying attention, is that the GBM cell at the center was swarmed by CAR-Ts and eventually threw in the towel and became green. So I'm now going to switch gears just as we finish here and touch on the rationale for the second antibody campaign, which yielded the lead nanobody that was actually characterized more deeply as CAR-T cell in the paper. And I think I'll eat my own words a little bit in terms of nanobodies and their favorable properties. I suppose because one of the problems we encountered with the best nanobody from the first campaign, MKRO20, was that although it had really great properties as a single domain antibody monomer, it didn't really like to be fused to other proteins like the FC region. We had a lot of trouble manufacturing that molecule due to low yield and precipitation. This is actually the only nanobody I've ever seen behave badly in this respect, which I suppose makes it unique and therefore probably priceless. I don't still have a very good explanation of this phenomenon. And it was only by going back and forth with Chirayu and his team on the early CAR-T results that we started to re-examine some of the other data, such as the flow staining shown here. And we eventually concluded that all the signs pointed in the same direction, in that the original antibody seemed to lose its favorable properties in fusion formats, including as a CAR-T, possibly due to low expression or aggregation. And this led to unacceptably variable and sometimes disappointing binding and efficacy data. So just to round this all out, I think the choice of the final lead was a really good example of how working with a multidisciplinary team and looking at the data collectively can really help drive things forward more productively. So in the end, the best nanobody and the best CAR-T, this was based on an antibody called XRO, was characterized further in Singh lab using cell line-derived xenograft models, first of recurrent glioblastoma. So again, this CAR-T cell showed the most potent in vitro cytotoxicity in co-culture assays with tumor cells. which could also be observed in their activated phenotypes, high levels of cytokine secretion, which aren't shown here. Anyway, in this model, 4 days after implanting recurrent glioblastoma brain tumor initiating cells in the frontal lobes of immunocompromised mice, 500,000 CAR-Ts or untransduced T cells are delivered as a single dose intracranially. And what you can see here is that treatment with the ROBO1 CAR-T cells clearly restrict or repress tumor growth and led to about a doubling of median survival from 35 to 75 days, with 3 of the 8 mice here that were treated with the ROBO1 CAR-T achieving long-term survival beyond 80 days. So that was amazing in itself, but some of the bright minds at Singh Lab had the great idea to ask the question of whether the CAR-T might be efficacious in other treatment-refractory invasive brain cancers. So the key to these additional studies was the surprising and exciting discovery that in addition to its known expression in some regions of the normal brain, ROBO1 was expressed at relatively high levels in patient-derived cell lines from adult lung-to-brain metastases, as well as in relapsed pediatric group 3 medulloblastoma. So these experiments had a similar setup, and the ROBO1 CAR-T was first demonstrated to exert very potent cytotoxicity in co-cultures with lung-to-brain mets and medulloblastoma cell lines. And as in the recurrent GBM model, a single intracranial dose of 500,000 CAR-T cells more than doubled the median survival compared with the untransduced T cell control. And even more remarkably, led to an apparent cure in the mice bearing relapsed medulloblastoma xenografts. So I think the overall significance of these results is that they point to our role for ROBO1 that goes beyond just invasion and migration of recurrent GBM. So this links this target to other invasive and migratory tumors of the brain. And while the extent of the biology of the PTP4A ROBO1 axis may vary in different tumors, this awaits further study. The nice thing about CAR-T cells is that sometimes the expression of the target at decent levels is really all that's required for efficacy. And I think the data here support a more general relevance of the ROBO1 CAR-T cell as immunotherapy for patients with different types of invasive brain tumors. So I think that's all I had, and I think I'll, at this point, hand things back to Sheila for question period.
Thank you so much, Kevin. And, it was amazing to learn that much about antibody engineering campaigns done with such high professionalism. And it helped us all really understand more about our own therapies and their efficacy. So that's the whole story. And I want to thank Jai and his team for inviting us to present this work. Again, I think Chirayu summarized about 4 years of mechanism work in a few slides, and the same thing with the antibody campaigns. But in the end, it was really rewarding to be able to bring this to completion. And Jason and I just came back from a GBM workshop organized by the Mark and Sontag Foundations in Chicago. And I think every other person was asking us when our RoboCAR-T is going into patients. So that's our next job.